Thanks everybody for joining today at the MIA Lightning Talk session. We have 5 speakers today who will all speak 10 minutes, and then we have 5 minutes for questions and a short break between and sometime after also if you want to follow up more. Yeah, the talks will be on the whiteboard. If you go much over time, I'll try give you sense. Yeah, anybody online, we also follow the Zoom chat. So if you don't want to speak up, you can write your questions and I'll forward them to the speaker. Yeah, and this is recorded. Yeah, thanks everybody for coming. And yeah, looking forward to the first talk now, which will be by Phillip Schneider from MGH. Who will talk about mixed integer linear programming and metabolic engineering. Really looking forward. Thank you.
Yeah, thanks for the introduction. I'm Phil. I work in the epigenetics group of Bob Kingston over at MGH. But I'm going to talk today about my PhD project, which I did in one of the Max Planck Institutes over in Germany, that was centered on constraint-based metabolic modeling. So I come from a metabolic engineering systems biology lab. So I assume that most of you are probably unfamiliar because this is very focused on, here you guys are more focused on medicine, medical applications. So to give you a bit of background, my former lab was more interested into modeling metabolisms, understanding metabolic diseases, and most of all, engineering microbes as bioproduction hosts to produce bulk chemicals, dyes, fragrances, and so on. So the centerpiece of my PhD thesis was constraint-based modeling and how we can use mixed integer linear programming and linear programming to engineer metabolisms in order to serve us in certain ways. So to dive into the basics, let's assume we have a very simple metabolic network with a substrate that is taken up and turned into metabolite A, which is turned into B via different reactions, and then B could be a biomass precursor, and then A could be also transformed into a different product. We can, so the arrows here are the different reactions that do the turnover that we can name. We can numerate 1 to 5, and now how constraint-based modeling works is conveying all this entire thing in a simple system of linear inequalities that we can write as G multiplied by R, smaller equals lowercase g. And this simplification, includes 2 things. One is that all metabolites within our system, that could be as our organism here, are in a steady state. So we are only able to model exponential growth or steady flux distributions within a cell. And the constriction here is that we cannot, the limitation is that we cannot model any transient stages, at least not with this very simple equation here. It does not add any genetic constraints, does not add any thermodynamic constraints. We can add them manually, but this is the very simplest way to look at it, and this is very powerful because this is a very simple network, but we can put up to thousands of metabolites, thousands of reactions in there, and this form allows us to apply different linear algebra tricks. So first, for very simple analysis, we can use we can just solve this equation, right? We can measure certain rates. Let's say we measured R5, we measured our growth rate. We can determine what our substrate uptake is. But more important than that, we can use just linear programming to look at what an optimization just based on a stoichiometric will look like, stoichiometric network will look like. So if we want to do the biological objective, that would just be maximize R4, or our engineering objective could be maximize R5. and both subject to this inequality system. So that is pretty nice for the analysis part. For the engineering part, we can now harness different other theorems. Let's say we have a metabolism and we want to see minimum failure mode. So what is the minimum set of reactions that have to crash in order to make the entire system crash? So we can, so our problem reads something like, what is the minimal set of knockouts, for instance, that lead to this guy here having no flux states with growth. And in order to do that, and in order to write this, we need to kind of find an optimization problem that makes our initial problem infeasible. And something that comes in very handy for this is dilemma of Farkas. Because it constructs a pair of two problems. One is a primal problem that has this form, and the other one has this form. And so the lemma now states that exactly one of the two has a solution. And as you can see, so our primal problem here has this form. We can now construct another one and link variables and constraints into this to our initial reactions. And by solving the Farkas dual, we'll find so the same exact interventions in our network that make the Farkas dual feasible are exactly the same that will make our primal infeasible. So it's a way in which you can take any problem, transform it, and see what are minimal interventions that will make your system infeasible. A second thing to exploit here is to harness is LP duality. So you can also very often in metabolic engineering you face kind of a conflict of interest between the biological goal or biological selection of fastest growing strains versus strains that produce what you're interested in. If you look at this, so the solution space of this reaction rates that are possible are convex polyhedron, so you can project it just down to fewer dimensions. Let's say you just project this down to a growth rate versus your production rate. And very often you will find something like this. So all these are possible solutions to the initial problem. There are some with like less growth and some production and so on. But just by selection, just by letting them grow, you select for the fastest growing phenotype, and that is one usually that does not carry any production. So now you want to come up with another maximization problem, with a nested maximization problem, because the engineer is interested now in maximizing production rate, knowing that the biological objective is maximizing growth rates. And then, of course, the model needs to hold. And we want to find a minimum set of interventions that we just name K. And these can be represented in binary variables. So LP duality allows us to take this nested optimization problem and make it all into one primal problem, and bring it essentially into this form, where some of the variables are binary variables. And by solving this, we can reshape the solution space to something that looks like this. So at some point, you will have a selection. Your selection for maximum growth will select for a phenotype that also has some positive production. Yeah, I hope I'm within the 10 minutes. Okay. yeah, so that's basically what I worked on. And so at the end of my PhD thesis, I put this all into, so there were different projects I worked on, but essentially this part here is something that I put into one big Python package that is available, which is called strain design.
How well does this framework handle the case where you don't know the network fully?
If you don't,
if there's like some other, if there's like a C there, but you don't know that it's there.
well, you have to assume that it's complete in order to model it completely. I think it's, well, if you, so if it's incomplete and you enter all your reaction rates, and then you try to solve it, and then it's like overdetermined, that could be an indicator that you have that there are like unspotted niches in your metabolism.
Maybe like a brief follow-up to that. Is there estimates or like any approach to how one could estimate like how correct like these networks and metabolomics are, like what fraction of reaction might be missing? Or
yeah, I think they're very well rather descriptive, because the people that build up these models usually test them and then fit them to their data. But I think there's still some beauty in just looking at the stoichiometry itself, and it leaves like a. So there's one technique that's called flux variability analysis, and it does not really pinpoint down a specific rate for each reaction, but you get the boundaries. So you say with this uptake and this growth rate, reaction X can only be in that range. And then afterwards, you can put on thermodynamic constraints and kinetic constraints and try to narrow it down to like a more realistic value. But yeah, often this, well, it's called constraint-based modeling, but very often it does not constrain it like a lot. And there's degrees of freedom in a solution.
And if you look at the molecular bioliterature, like, do people still find new enzymatic functions? Right, because I guess that would be an indicator also, yeah. Right? Or if that saturates, then
I think new models are definitely coming up. I think for the base models like E. coli, I don't think anything has really changed. I think the largest model might be from 2017, and they did not really make any. Yeah.
Thanks. Yeah, so next up we have Kai here from this floor and from Schmidt Center, who's a postdoc fellow who will talk about an optimal transport approach to single-cell data integration. Looking forward.
Hi, everyone. My name is Kai, and I'm a postdoc at the EWSC. And today I'm going to talk about my previous work, single-cell data integration with optimal transport. Okay, so in many cases, we have to integrate different data sets, probably from different modalities or from different batches. And by integrating these data sets, we can probably have like a whole picture of the data or the process. For example, like if we have data set X. Yeah. Thank you. If we have data set X and probably data set Y. For example, if we assume that if there are some cell types in data set X, for example, like this well, and some historical cell types that can be distinguished in data set X, so we can find a very easy classifier, but probably in data set Y, we cannot find such a good property. For example, like the mixed in the data set Y. However, if we have another cell types that can be distinguished in data set Y, that mixed in the data X. So by integrating the two data sets, probably we can find the 3 cell types if we can have a good latent space for these two data sets. So let's assume that there is a gene set in data set X. Here is gene set. And another gene set in data set Y. Probably they have some like common genes among these two data sets and some data set specific genes. So let's call this part the X specific genes. And this part the Y specific genes. And here is typically the common genes. So many existing methods, they only use the common genes for their integration methods. But like the specific genes also maybe provide more information about the cell types or the cell states. So how to utilize all of these genes, including the common and specific genes, is what I'm going to talk about. So here, there are some like examples. For example, we have morphage data as well as the single cell RNA data. They share some common genes and they have their specific genes. And maybe for other spatial transcriptomics data like Xenium data and cosmics data, they also have their unique gene panels, but they share some genes and specific genes. So let's have some like definition. For example, here for the X specific genes, we can find some highly variable genes to form a matrix. Let's donate it as XS. This is a matrix for the X specific genes, and for the Y specific genes, we also have some highly variable genes that donate as XYS. And for the common genes, we have two matrices, the X common and the Y common. So here we have 4 matrices here. We have 4 matrices. So how to use this 4 matrix for the integration? I'm going to introduce the model here. For example, this one is the X common. It's the common genes from dataset X, and we have the common genes from dataset Y, YC. The row is the gene, and the column is the cell, maybe. And their genes are exactly the same because they are from the common genes, and the number of cells may be different. So we input these two into a variational autoencoder. Here is the encoder. Let's donate it as C, and from this encoder, we can have a latent space, right? Have a latent space for the cells. And because it's a variational autoencoder for each cell point, for example, cell point xi, we can have like a mu, the mean vector and the standard deviation vector for xi. And a point from y, this at y, we also can have like mu and the sigma. And we use reparameterization to get like z for x and y, and then we use a decoder. It's a very standard variational autoencoder to decode both xc and yc. Okay, so it's a very basic structure, and let's write its like loss function first. It's we want to like maximize loss of the evidence lower bound equals to like expectation of like oh sorry, let's call this decoder phi. So given state x, we use the encoder to get a latent variable z, and we want to minimize such a log. Given the z, we want to reconstruct the x. This is the first term, and the second term is the KL divergence of the encoder. Given x, get z, and we want this one to follow a Gaussian distribution. This p is a standard Gaussian distribution. So this is a basic like variational autoencoder, and here we only use the common genes. So how about the specific genes? here we introduce a very simple way to incorporate the specific genes. Probably there are some like other ways. Here we want this latent space to decode not just the common genes. We want it to also decode the specific genes. This one is we donated phi x. We want it to decode x, the specific genes in x, and we want another decoder to decode the ys. So input the common genes, we want the latent space to decode both common genes and specific genes. Here also we have to add 2 terms to this loss function. That is like trade-off parameter, and like we want to add another term phi x. Given z, we want it to reconstruct x as, and given z, we want to reconstruct ys. Okay, so here is the loss for this autoencoder. But because there are some inherent differences between these 2 data types, and here we also introduce the specific genes which will further introduce heterogeneity in the latent space. So we have to employ some like distribution alignment loss to like make this the distribution from these two data sets to be similar. We have several choices. For example, we can use generative adversarial neural network, or GAN, or the maximum mean distance, or MMD, or optimal transport. So finally, we choose optimal transport, because if we use the GAN or the MMD, that means we hope the distribution to be exactly the same in the latent space for the 2 data sets. So they are fully alignment. But if we use optimal transport, we have a variation that is unbalanced optimal transport, which we can have like some data set specific cell types in X that we don't want it to align with any cells in Y. So this is the reason why I choose the optimal transport. So here the problem is how to calculate the Wasserstein distance between any data points before we calculate the optimal transport. So here, like in the latent space, actually we have 2 data points here as example. They are like follow a Gaussian distribution. So there is a theory that for 2 Gaussian distribution, maybe the mean is mu x and the covariance matrix is sigma x, and another Gaussian distribution mu y, sigma y. The Wasserstein distance between these 2 Gaussian distributions actually have like closed format, like the Wasserstein distribution, like let's donate this as px, and this distribution is py, is equals to the mu x minus mu y square plus trace sigma x plus sigma y minus 2 sigma x. Okay, this is the Wasserstein distance between any Gaussian distribution. And when the covariance matrix to be diagonal, that means this sigma can be right as standard deviation. Well, this sigma is like it's positive and it's a vector. Then the Wasserstein distance can be right as very simple way, mu xi minus mu yj square, plus sigma x minus sigma y. So here, like we have this variation autoencoder and the latent data point, like they follow this situation. So we can calculate the Wasserstein distance between any 2 data points here, for example, xi and yj, as well as if we use like I and J, we can calculate like this one to assess the distance between data point X and data point J. So after we get this Wasserstein distance between any data point, we can calculate the optimal transport. We can get a optimized optimal transport by find optimal T that using this Wasserstein distance. This is the inner product, and we can find another like entropy regularization term and plus unbalanced term. This is the trade-off parameter, and yes, we can use KL divergence. With this, the column, the sum of the columns follow a uniform distribution, and the sum of rows follow a distribution, a uniform distribution, and the sum of columns follow another uniform distribution. So here we found we can iteratively optimize these two problems. First, if we fix the variational autoencoder parameter, we can calculate optimize optimal transport T, transport plan T. And if we fix the optimal transport T, we can optimize C, where C comes from mu X and mu G and sigma S and sigma G, where mu X and sigma, mu and sigma are the outcome of like this variational encoder. So we keep optimize these two like problem, and we can get final convergence. Yeah, so this can be solved using a very typical optimal transport solution. Yeah, that's the whole method. Thank you.
Maybe if you want to comment on how you think about applications of this outside of data integration, because I guess you can do a lot of things with optimal transport, right?
Yeah. Actually, it's like it's just like MMD, or again, it's like distribution alignment loss kind of to compare the distance between two distributions. So we can apply it to a lot of other applications. Yeah. But here, maybe probably one possible idea is that we can like incorporate like the spatial information into this framework, because here we only use the gene expression. Yeah, but we can use the neighboring information.
I have a quick question about the identifiability of the model. Because I guess there's a few different things going on, right? If you change the autoencoder parameters, then you can reduce this loss by changing the autoencoder parameters as well as by changing the coupling.
Yeah.
Do you see that the same cells are matched if you train the model again from different seeds and things like that? Like how robust is the matching that you get from this?
Yeah, actually, the robust depends on the parameters in the variational autoencoder, right? If you change the seed, the initial parameters will be different. Yeah, but yeah, I think actually I haven't like investigated the variations for different seeds, but I think since don't matter, I don't know that, but I can do that later.
So next up is Bojan Karlas from DBMI at Harvard Medical School here across the river, who will talk about, actually also from ETH before, there's a number of ETH people here today, who will talk about data debugging with Shapley importance. So yeah, looking forward.
Cool. Thank you. So is this my slot or? Can I raise one more or not? Yeah. Got it. I think this should be fine. Yeah. Cool. So thanks. So hello, everyone. Thank you for the introduction. So my name is Bojan. I work as a postdoc at MGH and Harvard Medical School. And today I'll be talking about basically work that I did during PhD that I'm still sort of involved with. And recently it's getting published in iClear 2024. And it's called EaseML Datascope. And that's the work that I did, it's during PhD. And basically, the problem that we try to focus on here is we can call it targeted data debugging. So that means basically, very often when machine learning models experience poor quality, often those problems can be traced back to the errors in your data. However, the problem is that very often it's hard to find the exact data points that are causing the problem. And also among the ones that are causing the problem, we don't know how to prioritize which ones are more nefarious than the others, let's say. And so this work is trying to basically prioritize data points based on some measure of importance in order to help the sort of people who want to do data debugging, which is often a labor-intensive task, to prioritize their effort and do the debugging of parts of data that are more important. And we do that using Shapley value, which has been shown empirically to work pretty well. However, it comes with a problem, and that is it's computationally very expensive. And so this work is about trying to find ways to apply this to a machine learning pipeline in a way that's computationally efficient. So let me show you what I mean. So let's say we have, several data or maybe several tables. So this is our input data set. Maybe here we have also some label column. And so this input data set, we typically pass through some feature extraction pipeline that can merge and transform this data in various ways. And the output of this pipeline is featureized data set with features and labels. And then we pass this into some machine learning algorithm that produces a model. And then we take that model and we evaluate it using some quality metric, which can be accuracy, AUC score, or maybe a measure of fairness, equalized odds, or something like that. And also, for this, we typically use some validation data set. So what happens when, at the end of this, the model underperforms? So typically, the problem can be traced back to maybe some problems in the data. It can happen anywhere. Data errors. And what we're trying to do is basically help the user identify the most important data errors by computing some measure of importance. And the measure we chose, because it has been shown to work pretty well empirically, is Shapley value. So I'm doing a little bit bigger than I thought initially, but okay. Yeah, can I keep erasing? Yeah, I can. Sorry about that. Okay, let me write that part here. So Shapley value, how is that defined? We usually define it for a single tuple of the input data set. So PI is a member of the training data set. And let's say the size, we have n data points in that training data set. So it's defined as follows: 1 over n, and then there's a sum over all subsets of the training data set without Ti, multiplied by some normalization constant, and then something called a marginal error. And the marginal error is basically a difference in some utility, which is basically something that maps a subset of the data to some real value. For this pipeline, it can be defined as a metric. Sorry. Basically, it's a chain of operations on any subset using also the validation data. So pretty simple. The key issue here is, even though this is pretty useful, is that there are exponentially many subsets here of this data set. So this sum is basically impractical to compute. So that's not good. So in my work, we tried to basically find if we can discover some properties of these elements and approximate them somehow in order to make this computationally more efficient. So to do that, we made 3 approximations. The first one is the pipeline is something which we refer to as canonical. It's just a term we made up, but basically it means, it has a technical definition, but it means that it has a certain structure that lends itself to nicely computing Shapley. And the 3 categories of canonical pipelines that we identified was map, fork, and join pipelines. And this depends on, so these are the 3 types of general operations you can do with tuples in the input data set. Mapping, you can think of it just as mapping data points from input to the output. Forking is like every data point is somehow forked into multiple data points in the output. So think of data augmentation operations. And joining means that an input data the presence of an input data point is dependent on joining 2 or more input data points. And so that's the first approximation. The second one is that this model is the k-nearest neighbor model. And the third one is that the model quality metric has an additive structure. And what that means is that it can be decomposed into basically element-wise, into a sum or an average or some sort of aggregate over element-wise pieces between the predicted label and sort of the validation label. And if it had that structure, so if all these 3 conditions are met, then we can compute Shapley value in polynomial time. For the simplest pipelines, we can do it as fast as n log n. And for the more complicated join pipelines, we can do it. It's not ideal, but it is still p time, which was basically the result. So if I have time, I'd like to also show maybe like the very simple case. Yeah, OK. So yeah, let's go through the very simple case where we have a map pipeline and k equals 1. And here you'll basically be able to see like how we leverage the properties of the k-nearest neighbor algorithm in order to turn this formula into something that can be computed very efficiently. So let's look at this data set here. So we have x and y. Let's think of the simpler piece where we want to compute the Shapley value of some specific tuple Ti with respect to a validation data set that has a single data point. Okay. So how do we do that? So what we can observe is, let's say, for example, we have like T1, T2, T3, T4, 5, 6, let's say. And let's say this is I. So what we want to do is partition this sum or this set of subsets based on two properties. So first we can write just this piece we just copy. And then we split the sum based on two parameters. One is the size of the subset. And two is what is the most similar data example within that subset to the validation data set. So something called J. So if we look closely, how would the k-nearest neighbor algorithm make the prediction here? So first, it would basically sort all the data points based on similarity to the validation data point. So let's assume this is already sorted. So that means the T1 is the most similar data point. And the predicted label will always be Y. But if we remove T1, then the next one will be predicted. So basically, for every single subset, the topmost element will be the one that's going to be predicted. Okay? And what we care about is all the subsets where adding Ti is going to make a difference. So those are all the subsets that are below Ti. So that's why here we consider only this set of subsets. And the sizes of those data subsets are this much. So then we can copy this term. This is alpha. Sorry. And here, instead of this, we can use the marginal error term. So mind you, in all these subsets, Pi will be the most similar if it's in the subset. And if it's not in the subset, Tj, whichever it is, will be the most similar. So okay. And then we only need to multiply it by, because this is always the same. We partition basically the space of subsets. They all have the same property, that they all share the same marginal error. And we just need to multiply it by the number of those subsets. And in this case, if we look at basically, like, let's say this is J, and we have this many, how many subsets of size alpha are there in this part? And that's it. So basically, we transform this formula into this. And for the final step, which I'm going to skip actually a few steps, it can be further simplified. But you're going to have to trust me. Into this. So basically, to compute the Shapley value, we sort the data set, and we do one pass through the data set. We're able to compute something that's normally exponential, but because we're using the k-nearest neighbor model, we're able to do it in n log n. So this work has been done in my group where I did PhD. Mainly this part was done by my colleague called Mary Girl. This whole project was basically taking this as a starting point and then trying to study what happens when we make this pipeline more complicated. And when we make k greater than 1, how does this impact the computability? On top of that, we also implemented a Python package. For all that, you can find on this link, which is also a URL. There's demos, there's links to papers. Yeah, that's it. Thank you.
Thanks for the talk. Could you elaborate a bit on what you mean by data error? Is it like a labeling error or an outlier?
Yeah, it can be anything. Like it can be a label error. It can be a feature error. It can also be an outlier. Yeah. So any sort of, yeah, anything that's in your data that's kind of causing the problem to the model at the validation step. So an outlier is also an example.
Okay. Thanks.
Assuming that your training validation data come from the same distribution, then presumably there might be these so-called data errors in like the validation data set as well, right? So does the process of doing this debugging make your model more sensitive to any errors in the validation data set?
Yeah, excellent point, and that's exactly true. So we basically rely on the validation data set to be clean.
Okay.
Or whatever. So it can also, you can think of it also if a validation data set maybe has a different distribution slightly than the training data set.
Yeah.
We assume that the validation data set is what you want the model to drive towards. So
okay.
the data errors will drive you towards.
Yeah. Thanks.
Okay. I have a quick question. What happens if you, instead of trying to compute this utility over subsets, just do like leave one out? So like what do you gain when you estimate the Shapley value by in this way versus like just leaving one out from the whole?
Well, leave one out is kind of it is the simplest basically way to try to estimate the importance. It's also another way people do try to estimate importance. It works basically for if you want to just find one data point that is causing a problem. But think about it if you have like certain interactions between data points, like maybe there's some interdependency. Maybe a data point in its own, removing it doesn't help much, but removing that and multiple similar data points helps more. So that then all of them are somehow valuable, not just, but any single one of them is not that valuable. So basically, Shapley value manages to capture these kinds of interactions between data points. And it's been shown also empirically to do better at like at data debugging tasks.
Thank you.
Cool. Yeah, thanks.
So in this second part, we have two more speakers. First up is Chenyu here from Schmidt Center also and from MIT East, who will talk about removing biases from molecular representations. So looking forward.
Hi, everyone. I'm a PhD student at MIT ECS, co-advised by Caroline Wooler and Tom Yakula. And today I'm going to talk about our work accepted by ICLR 2024 about removing biases from molecular representations via information maximization, or the model's name is named as InfoCore. So the problem we are going to research on is to learn a good molecular representations. And with molecular structures, people will usually have some unimodal representation learning method, like purely based on the molecular 2D structure or small strings, based on some augmentations or other self-supervised learning way to learn the representation of the molecule. However, it can be hard if we only look at the molecular structure, because we can imagine that two different molecules can have very different effect in the cell. in real cells, even they have very similar structure. And it's hard to know which part of the structure of the molecule is having the biological effect to the cells and which is some kind of irrelevant part. So with this in mind, we aim to learn a multimodal representation where we can make use of the chemical perturbation data, for example, gene expression or cell imaging, which is, so for the gene expression, in our paper, we are using this connectivity map dataset. And for the cell imaging, we are using the cell painting dataset, both from the Broad. And we want to utilize this large-scale dataset because they can show when we add a molecule into a cell, how the cell will react to the drug perturbation, which will actually reflect the biological effect of the drug in the cell. And when using this biological data, one very significant challenge, as we know, is the noise and the batch effect in the data. And here we have a simplified graphical model for the effect of the batch effect in the data. And I will explain later why we call it a bias and why we want to remove this bias. So now assume that we have a drug structure, which I name it as XD. And also I have, this is called XG, which is a phenotypic profiles after the drug perturbation. So obviously there's an edge from XD to XG. And we also know that because these data are usually high throughput and they are done in batches, so there will be some batch effect in the data where we have another variable called XB, which is a batch number that the experiment is done. So this edge means the normal batch effect to this data. So this is a typical setting, which is not so hard, but surprisingly, what we found in the data set and what is a common issue in almost all the data set is that there's an additional edge from XB also to XB, which means that the drugs are not randomly placed in different batches. So due to some experimental reasons, they didn't fully randomly shuffle all the drugs towards different batches, and we can observe some similarity of drug structures within the same batch. And a simple experiment to show this is we train a simple random classifier predictor, which takes only the drug structure as input to predict the batch number. And in the held out set, surprisingly, in the two data sets that we are using, we find that the prediction accuracy is about 50% versus the random case should be about 1 or 2%. So this confounding issue is very severe. And now with this graphical model, we can see that this batch XB is actually a confounder between the drug structure XD and the phenotype XG. And this will make the learning an unbiased representation really hard because it will be hard to disentangle what is the effect caused by this confounder XB and the batch effect in the data and what is the real effect of the drug. So based on this, we propose a new method to solve this issue. First, it's about our optimization objective. So in normal multimodal representation learning, like CLIP, which is learning a joint representation between text and images, they are actually maximizing the mutual information between the, okay, by the way, we have two representations, which we call ZG and ZD, which are the corresponding learned representation from either the drug structure or the phenotype data. So a normal multimodal representation learning will learn to maximize the mutual information between the two latent representations of the two modalities. Then the latent representations will try to gather more and more shared information. However, because of the existence of this XB, we want to remove this bias. So instead, we are learning to maximize the conditional mutual information between the two latent representations conditioned on the batch number. And intuitively speaking, since we have a condition on the batch number, any batch-related features will not benefit to this objective, and they will be removed from the learned representations. However, to find a training objective for this conditional mutual information is not very easy. A naive way is to also, for example, sample the negative samples conditioned on batch, which means when we are doing contrastive learning, we are sample all the negative samples all from the same experimental batch. But because in experimental batches that we have here, one batch, there's only 200 or 300 drugs because they are using like 384 well plays to do the experiments. And that really limited the number of the negatives that we can sample. So instead, we derive an alternative lower bound that can, of this conditional mutual information, that can make use of all the samples as negative samples in the data, which the bound, I'm written it here as. So there are two important parts in this bound. The first one is so here I have this subscript 1 or 22K. And if you are familiar with contrastive learning, everything I'm named as 1 is the anchor and positives, and everything named as 22K is the negative samples. And in contrastive learning, they have an anchor, and intuitively speaking, they want the loss to make all the to make the anchor and positive close to each other and far away from all the negatives in the latent space. And the anchor and positives are paired data, which in our example will be the drug structure and the phenotype that are corresponding to each other. So there's also a symmetrical term for the other modalities. I'm just not writing it here to make it faster. And there's the second term. And there's also a symmetrical term, and plus some constant. So this formula looks really long, but it is actually very similar to the normal contrastive loss or same clear loss, if you're familiar with contrastive learning. So if we remove this part in the first loss term, it is exactly the same as the same clear loss. So we are putting, this is the positive, this is the similarity between anchor and positive, and these are the similarities between negatives and anchor. So we are putting positive and anchor closer to each other and negative and anchor far away from each other. But here, the important term is this additional term that we have. So we are actually reweighting the negative terms in the normal contrastive learning based on their posterior batch distribution XB based on the latent. So intuitively speaking, for a negative sample, if its posterior batch distribution is more similar to the anchor's batch distribution, then we will give it a higher weight. And if it is less similar, we will give it a lower weight. So if the batch distribution is more similar of the negative, then it have more information. It is more informative when we are learning the representation of the anchor because they have the same batch confounding information. And the only difference between them will be the non-batch related information. And the second term is just a simple classification loss when we are training the batch classifiers to estimate that posterior batch distribution. And finally, it's a constant. So this offers us a very clean framework to do the batch correction of these confounding issues. And it's a very simple lower bound of the conditional mutual information. And compare with the previous baselines I'm talking about that we just sample negatives from the same batch, where here we can actually make use of all the samples from all different batches, but give them different weights. Yeah, that's basically all the method. We test the performance in some simulation setting and also in this real drug perturbation data set, including a gene expression data set and a cell imaging data set. And it gave us better performance. And we also show that it's a general framework to remove the confounder in the representation learning that can also be applied to all the other scenarios when there's an existence of the confounder. And we show that it also performed well in a representation fairness task. And that is all. We have our code and paper online, and welcome to any questions.
Thanks for a great talk. So this is a naive question. Why do you want to maximize this mutual information between ZG and ZD? And what is the maximization over?
Okay, so first, the second question, the maximization is over the encoder's parameters. Like here, ZG and ZD are two learned representations, and we want to optimize the encoders so that they can maximize this conditional mutual information. And the intuition behind why we want to maximize the mutual information is that, so for example, we observe a gene expression. We don't really know which part is noise and which part is really caused by the drug. And when we observe a drug structure, we also don't know what substructure of the drug is impacting the cell and what are some irrelevant structures. But if we optimize this mutual information, we're maximizing the shared information between these two modalities, which we think, other than the batch confounder, if there's no batch confounder, then the shared part should be biologically meaningful, that it is really how the drug is having the impact on the cell to link them together.
Thanks.
Maybe continuing from that, does that intuition of maximizing mutual information continue to hold if you had other effects on the on the cellular state, right? Like there could be like another like biological process that confounds that, but not the drug state.
You mean there are some other variables?
Yeah, only to XG then.
Yeah, that is exactly why we want to why actually I think these two modalities can kind of supervise each other in the representation learning, because if there are other edges here, as long as there's no confounding with the XB, then if we're only maximizing this mutual information, then those other effects will not be learned in the representation.
Yeah.
And just, so you have to estimate you have some variation approximation to P of XB. Is that correct?
Yeah. So these two P hat are the variational distributions, and we select to approximate it as a weighted average of the single P hat, the P hat of XB based on single latent representations. And we do this first, it's because it is a simple approximation and can reduce a lot of computational cost. And also, there's another term. If we do this, we can show that it is bounded by 0. So we can reduce that to a constant. And also, it's a weighted average, so we can very flexibly adjust how much we want to rely on the existing, the anchor batch distribution, and how much we want to rely on the negative batch distribution. And if that weight, for example, is 1, that we are putting all weight on this anchor spatch distribution, then it will be the same as the sim clear loss. And in another case, we will be more concentrated on removing the spatch confounder.
Cool, thanks so much. So yeah, next up and last for today will be Sandeep, also here from the Broad Institute, who will talk about a spatial organization of tissues and self-supervised learning. So looking forward.
Cool. Yeah, I'm Sandeep. So picking it up. Sandeep. Yeah. Okay, cool. Yeah, I'm a third-year graduate student in the Harvard Bioinformatics program, co-advised by Fei Chen and Martash Babadi. And I'll be talking to you guys today about my project titled Connecting the Spatial Organization of Tissues to Cell State and Function Using Self-Supervised Machine Learning. So the original motivation for this project was the differential analysis of spatial transcriptomics data. So suppose you have two groups of samples, a control group of samples for spatial transcriptomics data and condition. And you want to summarize the contrast between these two groups of samples. This is a pretty fundamental approach in all of biology, comparing a control group of samples to a condition group. And it's a little challenging to do with spatial transcriptomics data. A first naive idea would be to reduce the data to a bulk aggregate, perform bulk differential expression analysis. But that would suffer from diminished statistical power due to A, neglecting spatial correlations in your data, and also B, importantly, the spatial heterogeneity in your data. It's often the case that, for example, if you have, let's say, a tumor labeled sample, the tumor may only take up a portion of the whole slide or image. So ideally, what we want to do is identify and aggregate the contrastive regions from our condition samples, and then compare the two groups of samples at this regional level rather than the sample level. So how can we do that? What we would ideally have in order to do this is a common representation space. So ideally, we have some sort of representer R that can take patches or local tissue motifs from our samples and then embed them in some unified representation space. So let's say we have a group of samples from control, another group from condition. Our representer would ideally pass all of a set of patches from these samples into some unified representation space such that we can compare the changes in tissue architecture between our two groups of samples. So the next question is, how do we learn this representer without any labeled data? It's seldom the case that we have underlying annotations for tissue structure in our spatial transcriptomics data. So a natural approach is to use self-supervised learning, which is a fairly popular paradigm these days. An example would be autoregressive modeling in NLP. And in computer vision, a common framework is called contrastive learning, as was alluded to in the previous talk. So one example of contrastive learning is, let's say you have a data set of, say, cats and chairs, but you don't know which images are cats and which images are chairs, but you want to train a classifier to figure that out. One idea is to take each image in your data set and perform two random augmentations of that image. So you can imagine maybe rotating the cat or cropping the cat. Now, both of these images are still cat-like because they came from the same underlying image. So you can tell your model to produce representations. Let's say this passes through a model. We can tell our model to produce representations that are similar for these two random augmentations and dissimilar to the other images in our data set. So in solving this task, the model sort of learns what it means for a cat to be a cat and how that's different from a chair, thereby essentially learning meaningful features of the data that can then generalize to other downstream tasks, such as classification. So inspired by the success of this approach in the computer vision space, we decided to take a similar approach to our spatial transcriptomics data. So this is a method that we call tissue purifier. And what we do is we essentially encode our spatial transcriptomics data as images of cell types. So let's say we have a toy data set with 3 cell types. So there's like circles and triangles and squares. What we can do is we can extract patches from these multi-dimensional images where each cell type occupies one channel in our image. So we can extract these patches. We can apply 2 random augmentations to them. Actually, sorry, I'm going to move this up here. So let's say one augmentation is, again, rotating the patch, and then another augmentation's dropping out a channel. Then we can essentially apply our self-supervised learning framework on these 2 random augmentations to train a model to learn representations of the underlying tissue structure in those patches. So what we do is we compare 3 different self-supervised learning frameworks. One is the framework I mentioned here, which is called SimClare. We also try a method called Barlow twins, but the one that we find that works the best is called DINO, which stands for knowledge distillation with no labels. And the premise for DINO is you have 2 models with the exact same architecture. And here the model is a convolutional neural network. So we have a student CNN and a teacher CNN. And these 2 models have the exact same architecture, but the teacher is an ensembling of past iterations of the student via an exponential moving average of the parameters of the student network. And so what ends up happening actually is that because the teacher is an ensembling of the multiple students, it is throughout training, it's consistently slightly outperforming the student network. So as you're training this model, the student is basically sort of chasing the teacher and sort of being guided by the representations of the teacher learns. So to formalize that, what we can do is we can pass the features learned by each model through a projection head. That's also the same between the 2. We apply centering to the features learned by the teacher network. This prevents any one dimension of the feature vector from dominating. Then we can apply a softmax to turn both of these feature vectors into probability distributions. So we have P of S and P of T. And then to encourage these 2 probability vectors to be similar, we can apply cross entropy loss. And we can backpropagate this loss to update the parameters of the student network and apply a stop gradient so that it doesn't update the teacher network parameters, which are only updated by the exponential moving average. And so what ends up happening is we're able to train this model to learn representations of these patches that represent local tissue architectural motifs in our data. So once we've trained our model, we can take our multidimensional cell type based image and we can draw a patch around each spot or cell type, pass it through our trained model, and then obtain a set of feature vectors that essentially encapsulate a summary of the local tissue structures within our entire data set of spatial transcriptomic samples. And so with those feature vectors, those representations, we can do several downstream tasks. We can query tissue motifs. We can cluster tissue motifs. We can also regress back the gene expression that we have and ask what genes have the most mutual information with spatial structure in terms of the arrangement of cell types. And finally, we can do a task that we call tissue distillation, which is basically training a classifier or a regressor on top of the learned feature vectors to sort of figure out what are the diseased portions of our disease labeled samples. And then we can do differential expression at this regional level, either in terms of figuring out changes in gene expression magnitude between diseased regions and healthy regions, or looking for genes whose spatial patterning changes between disease and control regions. And so in summary, we basically have this framework that allows us to learn representations of tissue structure, link these representations back to gene expression, and then in doing so, figure out how changes in tissue organization impact transcriptional programs. Yeah.
Thank you, Sandeep. Any questions? I have one to start off with, maybe. A number of the length scale of tissue slices that one gets right now sort of like covers aspects of tissues where you have anatomic regions like subtissue niches that are sort of homogeneous-ish on the anatomic level and maybe somewhat well mixed is a bad word, but like sort of well mixed in terms of like the cell type composition like within that niche, right? So I wonder, right, like conceptually, where do you see the differences between what one can achieve with self-supervised representations here and segmentation, for example?
So just to understand your question, you're saying if the entire tissue is, so to speak, homogeneous, can you use the representations?
No, it's necessary. Like let's say you have right, like maybe a tissue in which like a tumor is embedded, and then maybe there's like subclones of that tumor, and like these are like distinct tissue regions. If you have spatial transcriptomics, those may have like particular cell type composition locally, right, and like whatever particular profiles, like and you could try sort of like turn it into an image modality, and then run segmentation on it, which sort of like breaks the thing down into something a bit more tangible. But I guess it's more constrained than going into it with self-supervised learning. Maybe I don't know, maybe if you have ideas like how some of the downstream analysis would relate to things you would do if you had segmentation, and then would relate segments to each other.
Oh, I see. I think I guess maybe turning it around, one way that you could use the representations that the model learns is to actually perform that segmentation. You could take like the feature vectors of each local cell type composition and use that to sort of segment the tissue into different regions. And then you can look at those regions and then compare gene expression between them, for example, or changes in cell type abundances or other features of interest.
And like that, that works like sort of decently, that you get like segments.
Yeah, so the data sets that we actually tested and validated the model on are diabetic testis and aging thymus. In the testis, you have these like tubule structures that some of them, but not all of them, become disorganized during diabetes. And so we see that sort of with this, just this patching strategy, we're able to have the model sort of learn the boundaries of these tubule structures. And we can also learn the boundaries of different anatomical regions in the thymus, like the cortex versus the medulla, assuming that those anatomical structures are driven by differences in cell type compositions.
Yeah, okay. So because in the thymus you see like multiple nodules sort of, and they're all like cortex to medulla oriented.
Yeah, so like
right?
you have
like,
the thymus, you could see something like this. where like you have multiple cortex, sorry, medullary modules. And so assuming that you have a patch size that is large enough to sort of encompass the medullary nodules of interest, the models can learn how to sort of cluster or segment out those medullary regions from the rest of the thymus.
Yeah, that's nice. Yeah, thanks.
Mm-hmm.
Mine is, hi, thanks for the talk. So mine is a sort of quick one, but I'm just curious, since I don't know the literature in this area, but I'm interested in the topic.
Mm-hmm.
Do people normally do just these like rotations and simple augmentations, or is there something even more complicated? Because the concern, I guess, is that if the representation needs to be close with each other, if it needs to be just rotation insensitive, or invariant, is that sufficient to provide meaningful representations, or is there something?
Yeah. That's a good question. So like essentially the quality of the learned representations really depends on the set of augmentations that you choose. So let's say like for natural images, the set of augmentations that people commonly use are like rotation, blurring, random resizing crops, and several other things. For what we chose, I think like 5 or 6 different augmentations. So we do like rotations, we do flips, we do dropout of different like spots or dropout of different cell types. We also do multiplication by random intensity, Gaussian blurring, and we also do like random straight cuts across the tissue crops. And then we see that that set of augmentations helps us learn good representations.
So then still the patches that are the same tissue and close with each other in the representation space as a result. Have you observed this?
So what we see is actually we do see good mixing across multiple samples. So let's say this kind of depends on how homogeneous or heterogeneous your data set is across multiple samples. But if you look at the latent space of the model, it's not going to be that sample 1 is here and sample 2 is here. You'll sort of see mixing across the different samples, which is what we want to do. And some of that mixing is by choosing augmentations that make the model less sensitive to like sample specific effects.
Cool, thanks.
Mm-hmm.
Thanks. So following up, I guess, perhaps on David's question. So how important is the input dimension for this R? Because I guess depending on the granularity of what you're looking for, you have to choose this really carefully, right?
Yeah, exactly. Yeah. So I would say that's the main hyperparameter and perhaps even limitation of the approach, is that you need to figure out a patch size that is like corresponding to the length scale of interest that you want to study. So the way we did that for the data sets that we studied were just based on prior knowledge. Like for the testis, where you have these tubule structures, it's sort of straightforward to pick a patch that roughly corresponds to the size of a tubule. But we've thought about some automated ways to figure out those length scales of interest as well.
Have you thought about trying like neural operator type approaches that have more a physical notion of space as opposed to number of pixels?
I'm not familiar with neural operators actually, but that could be something good to discuss with chat after about.
So following the previous question, I'm asking about training classifiers on these patches. So imagine if you have a control sample and it have a disease sample, and I imagine if you're studying a tumor, then probably your tissue slice will contain some of the tumor's tissues and also some of the normal peri-cancer tissues that surrounds it.
Mm-hmm.
So I'm curious about how you add these classification, like the training labels, to these patches. Do you like add a normal tissue section, for example? You patchify the tissue sections where there is in the cancer patient or like the cancer sample, disease sample, but it's still normal, or do you distinguish between these?
Right. So what we do is we take our whole data set, we patchify it, and then we sort of separate it into like a spatially non-overlapping train and test set. And then so we train a regressor or classifier to predict the overall sample label from the train set of feature vectors that correspond to the patches. And so that train set's essentially going to be a noisy train set, because some of the diseased patches will actually be healthy. But assuming there's enough signal in that training set, then we can use that training set to predict the status of the patches in like a held out test set, essentially. Does that answer your question?
Yeah, thank you.
Yeah, thanks a lot, Sandeep.
Cool. Thanks.
Yeah, that concludes the MIA session for today. Thanks again, Philip, Kai, Bojan, Chenyu, and Sandeep just now for preparing the talks. And we see you all next week again.